CAO seminar tomorrow at 11 o'clock in room 109. We'll begin by Lokshin from Weizmann. So we're talking about the importance of dry intrusion of air-sea interaction, for air-sea interaction and weather extremes. I don't know if there's a geological survey lecture on Sunday. I couldn't seem to find a message from Emil. So if anyone knows, let me know. And so I think we'll move to our speaker today. So it's Dr. Anton Lokshin from Ben Gurion University. He did his bachelor's here at Hebrew University in geology and atmosphere. He then went on to do an MSc with Efrat, and then moved to Ben Gurion to do his PhD with Avner Gross and Daniel Palha. And he's now doing a postdoc abroad, and so we'll hear about his PhD research.
Yeah. So thank you very much. So I'm Anton Lokshin. I used to say a PhD student, but I received a PhD degree months ago, which I did under supervision of Avner, which is also here, and Daniel Palhan. And I'm excited to be here because I grew up in this institute, and it's a big honor for me to be here and give the talk in front of you. So thank you, Simon. And today we'll talk about the role of atmospheric dust as a global nutrient source for plants. And I will show you the work from the past 4 or 5 years, including my contribution of other students who worked on the project, which resulted in several publications. And I would like to begin by highlighting the huge amount of atmospheric particles that's circling in Earth's atmosphere. This animation made by NASA and to present 1 year of a global atmospheric circulation. Each color here represents different atmospheric particles. So you can see here the white color that represents eruption of volcanoes, so it's volcanic dust. The orange color represents desert dust, most of it in the Sahara Desert. And you can see how it crosses the Atlantic Ocean toward the Amazon rainforest and the Caribbean rainforests. And the green color represents fire ash and soot. Most of them are generated in the savannas of Africa and South America. And the main idea that I wanted to show here is the huge amount of masses that can move from continents and cross the oceans. And one of the interesting questions in ecology is how come the tropical forests, which are the most productive and biodiverse ecosystems on Earth, yet they grow in highly weathered soils with extremely low fertility. And when I say fertility, I mean to the primary limiting nutrient in tropical forests, which is the phosphorus. And there are several answers to this question, but the main answer is that tropical forests run on a very strict phosphorus budget, and they are very good in recycling the phosphorus within the system. So, for example, before the old leaves fall, they can resorb part of the phosphorus back to the tree, and the fallen leaves and branches rapidly decompose due to the high temperatures and humidity and rapid activity of microbes in the soil. So it is very efficient in the recycling of phosphorus with almost no losses, but there are some losses due to leaching and sorption of the free phosphorus to minerals in the soil. So for many years already it's suggested that there should be additional external source of phosphorus, and it suggested that it arrives with the Saharan dust during the dust season and deposited in the tropical forests. But there are two major questions remain unresolved. The first one, how does dust phosphorus enter plants, and how much dust phosphorus is reaching the ecosystem? But let's start at the beginning from the dust. And so the three most abundant atmospheric particles in the atmosphere are desert dust, volcanic ash, and fire ash. And these particles are enriched with the life-sustaining nutrients you can see in the green color from the periodic table: phosphorus, potassium, calcium, manganese, iron, and others. And each year, an estimated 2 million tons of dust is emitted into the atmosphere, with 75% deposited on land and 25% over the ocean. Yet most of the study on the impact of atmospheric particles focuses on oceans, because oceans are limited to phosphorus and iron, as you can see in these two figures. The purple color represents near zero concentration of phosphorus and iron. And atmospheric deposition is considered a critical nutrient source for ocean life because it supplies essential elements like iron and phosphorus that fuel algal productivity, and it can be pretty easily detected by remote sensing and reproduced in mesocosm experiments. So we wanted to take this idea of a blue ocean, a blue desert, and to apply it onto terrestrial ecosystems, because large areas of terrestrial ecosystems regarded as nutrient desert due to their extremely low availability of essential elements. This is an example of a global limitation of nitrogen and phosphorus. Red color represents nitrogen limitation, which is mostly prevails in the northern and cold regions, and phosphorus is the blue color. You can find the limitation mostly in wet and hot climates and geologically old regions. And one of the most important factors that affect the availability of nutrients in the soil is the pH of the soil. So this figure shows the pH values from 4 to 10, from acidic conditions to alkaline conditions, and then the availability spectrum of different elements. So if we look at phosphorus, its availability mostly unavailable below pH 6 in acidic conditions, and that can explain partially the strong limitation of phosphorus in the tropical forests. And if you look at the iron, manganese, copper, and zinc, they're mostly unavailable at alkaline conditions above 7.5, conditions like here in the Mediterranean region. So the question is, what is the impact of the deposited dust on terrestrial ecosystems? Now, we know that on the long term, dust contribute to soil fertility and it take part in building the soils. But what is the direct and immediate impact on plants and on trees is mostly unknown. And we think that there are two reasons for this question why it's unknown, because first one, the amount of the deposited dust is significantly lower than the bulk mass of soil. And second, it's pretty difficult to detect from where plants and trees took the nutrients, whether they took it from the dust or from the soil. So you need to apply geochemical tools to trace. Several studies from recent years show that trees are very good filters of the air because they can hold and retain atmospheric particles on the branches, on the leaves. And we're thinking that maybe leaves are not only holding the dust, maybe they can absorb nutrients from the dust through the leaves, because foliar fertilization is a common practice in agriculture by using chemical fertilizers, but the ecological connection between plant nutrition and dust is mostly unstudied. And if it occurs, so there are several advantages for the foliar uptake, because leaves are effective at capturing dust and holding it, and it reduces competition between microorganisms in the soil for nutrients, and there is no risk of the released nutrients binding to soil minerals, as it happens in the tropical soils, for example, making it unavailable to plants. And another trigger of our hypothesis was to look at this diagram that shows the trajectory, evolutionary trajectory from the green algae toward the modern plants during millions of years. While the modern plants have modern organs like roots they can use to absorb nutrients, so the green algae is fully immersed in water and it absorbs all its nutrients through the cell surface. And we were thinking that perhaps plant leaves, which part of them are developed from the green algae, retain the ability to uptake nutrients directly through their foliage, similar to algae. So to answer this question, we started with the greenhouse experiments, but before we collected and produced analogs of dust and ash. We collected dust from Negev Desert. We collected volcanic dust from Mount Etna from Sicily, and we produced fire ash by burning branches of pine trees. And we start our experiments by growing wheat, chickpea, and maize, and we starve them for phosphorus and their nutrition. Plants without phosphorus cannot grow, and after a very severe starvation, we applied atmospheric particles as a phosphorus source to see if there is a change in biomass. So at the first set of experiments, we added the dust toward the roots of the plants. And here are the results. So y-axis is the difference in biomass, x-axis is the different atmospheric particles that we added, red color represents the control group, and we didn't see any difference between the biomass. In the second set of experiment, we applied dust in a more natural way. This is a very starved chickpea plant. So we applied the dust from above, and we call it foliar application of dust, kind of mimic the way the dust arrives and settles. And this set of experiments showed a significant increase in the biomass of plants after applying 3 different types of atmospheric particles. And also we saw an increase in phosphorus content in all the groups, showing that the dominant way that plants can uptake nutrients from dust, or phosphorus from dust, is through the leaves. An additional experiment that did Maya Starr, a student of Avner, and Tamir Klein from Weizmann Institute, she applied dust on leaves of 3 trees, and she saw also differences in phosphorus. You can see the orange color, showing that this phenomenon also occurs in trees. And as I said at the beginning, it's pretty difficult to detect the source of nutrients, whether they took it from the leaves or from the roots, from the soil or from the dust. So we used 2 geochemical tools to understand the source. The first one is to look at the neodymium isotopes, and we used 2 distinct types of dust, volcanic dust and desert dust, which have significantly different epsilon neodymium values. And we detected the isotopes in the treated plants, and thus we were able to quantify and to trace the source, that it came or from the leaves or from the roots. And the second method that we used is to look at the rare earth elements, the REEs. And this method also commonly used in biochemical studies because elements or the sources of elements keep the unique pattern of distribution of the REEs. So we looked and studied the patterns in the dust and in the plants, in the dust in the plants and in the soil. And in addition, we looked at the ratio between the light and heavy REEs, the lanthanum and lutetium. So we saw the ratio in dust and in soil, and then in the control plants. And the treated foliar plants have a shift toward the dust, showing the source of the nutrients. And we did another set of experiments under elevated levels of CO2, thus mimicking the future climate, to see if the foliar uptake have any effect or changes as a result of it. So one of the phenomena at elevated levels of CO2 is that the biomass of plants expected to grow, and the carbon content also expected to grow, but at the same time, the mineral content or the nutritional value of plants expected to be lower in nitrogen, phosphorus, potassium, calcium, as many publications have showed, and they even said it threatens human nutrition because the nutritional value of plants expected to be lower. And there are 2 main reasons for this. The first one is simple dilution. If you take the same amount of nutrients and dilute it in a higher carbon content or biomass, so you will need to eat more plants to receive the same amount of minerals. And second is just downregulation of the roots in uptaking the nutrients from the soil. And so we grew plants under elevated levels of CO2. And as expected from literature, we also saw a significant reduction in the elements, nutrients in the plants. And when we applied volcanic dust, we saw a significant increase in iron, nickel, and phosphorus content. And when we applied desert dust, we saw also an increase in iron, nickel, and phosphorus content. And we suggested that maybe foliar fertilization may partially mitigate the expected nutritional decline. So we showed this phenomenon that it is significant and detectable in greenhouse experiments, and we wanted to go outside to see if we can detect and see this phenomenon also in a natural ecosystem. So we did an experiment in collaboration with Professor Marcelo Sternberg from Tel Aviv University. We did the experiment at LTR Mata in the downhills of Judea Mountains. This area receives dust from the nearby deserts. And we identified 3 naturally growing plants there, Marva, Gada, and Lotem. And we applied dust during 3 months, during the dust season. And we applied dust at 3 pulses to mimic the natural way that dust arrives. And to minimize the natural hazards like wind, rain, and wild animals, we built structures around all the plants. And after 3 months, we terminated the experiment, collected the plants, and analyzed them. And here are the results. So the brown color represents the control plants, and the purple, the treated foliar application of dust. And we saw a significant increase in the concentration of iron, manganese, nickel, and copper. And this pattern repeated itself in the 2 other species. A very significant increase in iron, manganese, nickel, and copper. In a parallel experiment that we did only on Lotem, we added dust to the roots area at the field experiment, and we saw also significant increase in iron and nickel, but in a much lower values, which showed us again that the dominant way of uptaking nutrients from dust is through the leaves. So what is the mechanism? Another PhD student of Avner, Natan Golan, he studied this phenomenon of foliar uptake from plants by growing several lines of chickpea and wheat plants, a lot of plants. And some of them were responsive to foliar application of dust, while others less responsive. And he saw that the most responsive lines have a significant increased concentration of trichomes. Trichomes are like hairs that are growing on the surface of leaves, and they are very good in holding particles. In addition, they can exudate organic acids from the tips of the trichomes, and the organic acids can promote the dissolution of insoluble minerals. So to summarize it up, so high trichome density, it helps in dust retention, exudation of organic acids lower the pH and promote the dissolution of insoluble nutrients. And the trichome bases, which you can see here in blue color, are suggested to be as possible pathways for dissolved ion transport across the cuticle, as they contain porous, permeable to ionic substances. And he also showed that the most responsive lines have a significantly higher exudation of oxalic, malic, and citric phosphoric acids. And these acids promote the dissolution of nutrients, insoluble nutrients. We also studied the chemical environment of plants in our field experiment. So the pH of the leaves was around 6, which is much lower than the pH of the soil, which was 8, alkaline soil. And we identified the organic acids that were exudated from the leaves, which is malic and citric. And to better understand the impact of the organic acids on the bioavailability of nutrients in the dust, we created an artificial foliar solution made from the organic acids that we detected. And we took the dust, we put the dust on a leaf-like surface and sprinkled the artificial solution and waited for 2 weeks, and then we measured the bioavailability of nutrients, and we saw a significant increase by several orders of magnitude in bioavailability of magnesium, manganese, iron, nickel, copper, zinc, which explains the increase. So the field experiment showed us a very important variable. What is the bioavailable fraction of each element in the dust through the leaves? So if you take, for example, a typical dust particle, about 2 to 5% of it made of iron, let's say 3%, and from the 3%, about 0.3% is bioavailable to plants through leaves. So we decided to look in a global scale where this phenomenon might be significant, and we did this work in a collaboration with Dr. Tom Goren from Bar-Ilan University. So first of all, we took dust deposition fluxes from the work of Koch et al. On annual global scale. Then we took a concentration of each element in the dust sources. And then we applied the number which we found from the field experiment, the actual bioavailable fraction. So we received this kind of map that shows the global annual fluxes of bioavailable iron, phosphorus, manganese, and copper. Then we wanted to compare and to see what is the fraction of nutrients that plants can take from leaves in comparison to fluxes from soil. So we took from literature a work of Ochoa Hueso 2023. He measured fluxes of different nutrients around the world, different points. So we compared our results with these results. So we divided our results, which you can see in the map, by the fluxes in the soil, and we received the fraction of nutrients contributed by dust via foliar uptake in an annual scale. So you can see here two examples for iron and phosphorus. So we discovered when we looked at iron two hotspots, the Mediterranean region and Western US. So these are very dusty regions. So the contribution through leaves can reach up to 70% there because of the very high dust deposition rates. But when we looked at phosphorus, we discovered this interesting point at the tropical region. And here the dust deposition rates are lower than the very dusty regions. But as you remember, the available phosphorus there is very low. Strong limitation of phosphorus, so even in moderate dust deposition rates can be significant and can supply up to 12% annually. I want to focus again on the Mediterranean region and say that most of the nutrients arrive in a few dust days. The average dust days in Israel is about 28 from literature. Thus, because it's episodic, dust annual comparisons underestimate its true contribution. So we did, instead of annual comparison, and per dust EVA comparison. So this is an example of iron. And bioavailable iron from atmosphere in a single dust event can supply more than 100% in comparison to iron daily fluxes from the soil via foliar uptake. And we did the same analysis for phosphorus, which we saw that again during dust event, the contribution through leaves can reach up to 100% in comparison to soil, manganese up to 5%, and copper up to 50%. So we successfully showed and published that we detected the foliar uptake phenomenon also in field conditions, natural conditions. And the global analysis showed us this point. It was a surprise, which reminded us about what I started from, the tropical forests. And we were thinking maybe the foliar uptake is a significant phenomenon also there. So we decided to do an additional field experiment in the tropical forest of Puerto Rico, because Avner works there for several years already. And we did the experiment in a collaboration with Dr. Tana Wood. She works at the USDA, and she's the head of the local tropical research station. And we think that doing the experiment there is a good fit, because Puerto Rico is one of the first islands that encounter the dust after it crosses the Atlantic during the dust season from May to September, and they experience severe dust days like we know from our region. And the natural pollution there is very low. They experience strong phosphorus limitation in the soil, which is also important, and it's managed by the USDA, so it's very convenient to work there. And last year we met Professor Jan Berenstoet from Wageningen University from Amsterdam, and he studies the impact of atmospheric deposition on the Atlantic Ocean. And during his sail during his expedition on a vessel, he collected dust, transatlantic dust, that was on its way to the tropical region, and part of it settles in the filters. So he gave us part of the dust which we used in the field experiment during last August. It was a pretty challenging experiment because we bring all the equipment with us, and we identified two understory trees that are common in the forest, and we built the structures again to protect the leaves from wind and rain and other animals. And we used two types of dust, the transatlantic dust and the analog from Israel that we bring with us. And if you're interested in our experiment in the field, in the tropical forest of Puerto Rico, you're welcome to read the paper that Avner wrote and published in Musaf Haaretz. And parallel to the foliar application experiment, we also ran an incubation of dust and local soil. We collected local soil from the forest. The soil there is very acidic. The pH was around 4.5. So we wanted to see if we add dust to the soil to increase the phosphorus flux. And we used the membranes which were also used at the work of that measured the fluxes of nutrients in soil. And they kind of mimic the way that plant roots absorb nutrients. So they can measure flux of ions. And the main idea was to compare dust through soil and dust through leaves. Now, one of the big challenges in field experiments, and especially in tropical forests, is to detect changes in phosphorus, because phosphorus is a very highly mobile nutrient, and it rapidly redistributes. It moves away from the receiving tissue to the other plant parts. So when we did the greenhouse experiments, we were able to take all the plants to the laboratory, weight it, and to detect the differences in phosphorus, but we cannot bring the tree to the laboratory. So instead of this, we used iron as an anchor to calculate the phosphorus, because iron is highly immobile and it accumulates in the receiving tissue. And we saw a significant increase in the concentration of iron in the studied leaves, the trees. And we took advantage of the experiments that I mentioned that did Maya Starr. She applied dust on the tropical tree, on the leaves of tropical tree, and she saw the differences in phosphorus and iron. So we took from there the conversion factor, and we applied it in our experiment to detect increase in phosphorus. So the incubation experiment of soil and dust showed an increase in phosphorus. This is soil, local soil without the dust, and this is the soil with dust. So this is the phosphorus. And also the foliar application experiment showed a significant increase in phosphorus, but the amount of dust that we applied and the surface area that we applied was different, so we needed to normalize the results per 1 gram per square meter, and here are the results of dust phosphorus uptake efficiency after the normalization. This is the foliar pathway and the soil pathway, and again we see a big difference. So why is the big difference? When phosphorus released from dust to soil, so it rapidly sorbed to iron oxides, which are common. It is a common mineral in tropical soils due to long-term weathering. And the sorbed phosphorus on iron is not available to plants. And when we did the analysis to release the phosphorus from iron, we see a significant release of phosphorus from the iron. In addition, we collected the dust that was lying on leaves of the studied trees, and we saw that its bioavailable fraction of phosphorus of the dust that was lying on leaves was significantly higher than the dust that was not exposed to the mildly acidic environment of the leaves. So, based on the results we got and the data we took from literature, we wanted to calculate fluxes of phosphorus in tropical forests of Luquillo and Ponto Único. We know that the plant phosphorus demand and uptake from soil is about 15 milligrams of phosphorus per square meter per month. We know from literature that dust supplies during the dust season about 8 milligrams of phosphorus per square meter per month. And according to the classical view that ignores the foliar pathway, the foliar uptake, it contributes about 0.015 milligram phosphorus per square meter per month. And if we take into account the foliar uptake, it contributes 1.29 milligrams of phosphorus per square meter per month. And if we do a percent contribution of dust to plant phosphorus budget, so from roots it's pretty negligible at 0.1%, and from leaves it's 8.6%, which is a very significant number. And this brings us to what I think one of the most important insights from this work, that the trees themselves play a very critical role in capturing the dust and bring its nutrients into the ecosystem. Because the canopy of the trees are very effective, much more effective than open surface in capturing the dust due to the high surface area and stickiness of the leaves and the complex structure. The usual way, or the standard way to understand to quantify how much dust arrived to the tropical forest is using buckets on open surface, which probably underestimates the true amount that arrives to the forest. And we think that if we'll be able to quantify how much dust the canopy can hold and can catch, it can change, together with the foliar pathway, it can change the way that our understanding of how much phosphorus arrives to the tropical forest through the atmosphere. And usually the assumption is that the surface area of the leaves, or the LAI, leaf area index, is higher 4 to 6 times than the surface. And if we take only the 4 assumption, which is the conservative assumption, so we can multiply the 8.6% by 4 and receive something like 34% contribution. And if these numbers are correct, we're looking at a different story. And that is the question that we're trying to answer in these days through experiments with small trees on the leaf of the roof of Ben Gurion University, and also in Laha Forest. We try to understand how much dust the canopy of the trees can hold and bring into the ecosystem. Maybe they can select dust particles that are enriched with nutrients, or there are different sizes that are preferable to the canopy. And if the experience will be successful, we can replicate them in tropical forests of Puerto Rico with real big trees, which I hope will be my postdoc project in a collaboration with colleagues from the US. And finally, I think that the foliar phosphorus uptake should be added to the air system models because it will improve estimates of plant available phosphorus and net primary productivity and carbon storage. In addition to improve air system models projections of CNP dynamics in the future of elevated CO2, because a higher net primary production is expected together with increased demand of phosphorus. And it's important to remember that tropical forests are a global climate tipping point, and elevated CO2 and drought are expected to constrain root uptake from soil, making the foliar pathway an increasingly central pathway for plant phosphorus acquisition. And the take-home message, I think what we're trying to do is move from a soil root-centric view to a vegetation leaf-centric view of plant nutrition by desert dust. And this phenomenon is especially significant in nutrient-poor, dust-affected ecosystems, such as the tropical forest. And thank you very much. If you have any questions, I'll be glad to answer.
In the tropical forest, the canopy is such that actually only the upper part of the canopy can receive the dust, because all the other plants are actually sheltered by the canopy. So how the dust arrives to the other trees?
To other trees?
The lower part. You know?
Yeah, so part of the dust is with the rain, it settles down toward the smaller trees and also to the soil through the withdrawal.
I think that you should make an experiment that really examine what is the difference between the canopy, the upper part of the canopy, and the lower part of the forest.
Yeah.
Because most of the small trees in the forest and the jungles, was growing there, so they are sheltered.
Yeah, correct. This is one of the plans to measure the dust deposition in the upper part and the lower part of the forest, but the atmospheric particles sometimes are very, very small, so they can penetrate the canopy and settle on the understory tree, I think, also.
And I also have a suggestion. My suggestion is to do experiments in the mangrove forest, because in the mangrove forest you don't have the issue of having a soil, because they are growing on the sea. So you can see directly, without having the interference of the soil, what is the behavior of the mangrove.
Thank you.
And you have them here inside.
First of all, an excellent talk.
Thank you.
I really enjoyed it. And I would like to ask you whether you also looked at not only nutrients, but toxic elements that are transported by dust. Lead, cadmium, and others.
Yeah.
Because,
Yeah, so it's a good question because most of the studies and papers on the leaf uptake, they concentrate on the toxic elements and how the toxicity of heavy metals affect the trees and the plants. But we focus on the, I also saw increase in other toxic elements. But as we showed in the greenhouse experiments, that the beneficial impact is higher than the toxic because we saw an increase in biomass and an increase in phosphorus fluxes. So it's a debate. There is always some harmful effect and also a beneficial effect, I think. Yeah.
Thank you very much. It was really clear, even for someone that don't really understand things.
Thank you.
I think so. You know how long does it take for the plant to take the nutrients from the dust or the leaf?
I think it's a couple of days.
So I think that the major difference between the tropical forest and the Ben-Gurion roof, for example, is the fact that in tropical region you have a lunar cycle of rain, right?
Mm-hmm.
So I think it would be washed out from the leaf quite quickly.
Mm-hmm.
So I don't know, maybe it's something to consider. How quickly can they really uptake the nutrients before it is washed out?
Yeah, it's a very important point because it's a big challenge in tropical forests to understand. I know from literature, if I'm not wrong, that about 50% of the dust arrives with rain in the tropical region. So we still don't know how it affects the dust. Maybe it, because rain is partly acidic, so maybe it dissolves part of phosphorus and it helps to promote its entry to the plant. But we need to do experiments with rain and wet deposition to understand the dynamics of it. So we'll have to do it.
I just want to follow that up. So, I saw you mentioned very briefly on one of the slides dew and the role of dew formation. But that would presumably be something that would also mediate, particularly in this region, whether or not that dust is bioavailable or not. Is that something you considered?
The contribution of dew?
Yeah.
I know that Marcelo Sternberg is suggested to do experiments with dew and dust because I think it has a very big contribution. It's a good point. It's a good idea.
Any more questions? Okay.
What happens to the light availability when the leaf is covered?
Yeah, so it for sure affects the photosynthesis and the ability of plants to do photosynthesis and gain carbon. At my first experiments, I added a lot of dust and I killed several plants because they were unable to do photosynthesis. And I also measured the photosynthesis rates, a different amount of dust. So there is like a sweet point where plants can hold. When I did the manual application of dust, of course, I exaggerated and I sometimes killed the plants. But in the natural conditions, I think they know how to hold enough dust that will help them, and in the same time to release the dust that it will not affect the photosynthesis. But yeah, I saw a reduction in photosynthesis when we measured the. But again, the beneficial effect is, I think, is higher because it increased the biomass and the phosphorus.
Okay, so let's thank our speaker.
Thank you.
Before we